Cognitive decline is one of the most feared consequences of aging, yet centenarians, those who live beyond 100 years, often defy expectations. A striking proportion of these individuals maintain functional independence, emotional stability, and in many cases, preserve memory and executive function well into extreme old age. This phenomenon, sometimes referred to as neuroprotection in longevity, is not a simple result of genetic luck or passive aging, but reflects a complex interplay between biological, environmental, and psychosocial factors that support resilience of the aging brain. Understanding these mechanisms may not only illuminate the biology of long life, but also offer strategies to promote cognitive health in the broader aging population. One of the most remarkable findings in longevity research is the comparatively low prevalence of clinical dementia among centenarians, especially in populations with high-quality aging trajectories. Although age is the strongest known risk factor for Alzheimer's disease and related dementias, studies in centenarian cohorts, such as those in Okinawa, Sardinia, and northern Italy, have revealed that a sizable number of individuals reach extreme ages with no significant cognitive impairment. Even when some degree of neuropathology is present, as in post-mortem studies of centenarian brains, the clinical manifestations are often surprisingly mild or absent. This paradox suggests the presence of neuroprotective factors, either delaying the onset of neurodegeneration or compensating for it functionally. Among the most studied of these factors is brain-derived neurotrophic factor, BDNF, a protein crucial for synaptic plasticity, neurogenesis, and neuronal survival. BDNF levels typically decline with age, correlated with cognitive impairment and brain atrophy. However, centenarians and their offspring often show higher circulating or brain levels of BDNF compared to age-matched controls, possibly due to genetic variants or lifestyle patterns such as regular physical activity and nutrient-rich diets. BDNF enhances the brain's ability to rewire itself in response to injury or loss, providing a molecular basis for cognitive resilience even in the face of accumulated cellular damage. Another key contributor to brain health in the oldest old is the suppression of chronic, low-grade inflammation, a condition often referred to as inflammaging. Pro-inflammatory cytokines such as IL-6 and TNF-A are strongly implicated in neurodegeneration, blood-brain barrier dysfunction, and synaptic loss. In contrast, centenarians frequently exhibit a favorable inflammatory profile characterized by lower levels of these harmful cytokines and higher levels of anti-inflammatory mediators like IL-10. This immune balance may help preserve neural tissue and reduce oxidative stress within the brain, acting as a buffer against both vascular and neurodegenerative pathologies. Vascular health is also central to neuroprotection. The brain depends on a constant and tightly regulated supply of oxygen and nutrients, and even subtle disruptions in cerebrovascular function can lead to cognitive decline. In long-lived individuals, vascular integrity appears to be better preserved, with reduced incidence of atherosclerosis, microbleeds, and white matter lesions compared to younger elderly peers. These protective effects may be partly genetic, but are also influenced by lifelong cardiovascular health, lower blood pressure variability, optimal lipid profiles, and low glycemic burden all contribute to a stable cerebral environment. The resulting hyperfusion and reduced oxidative damage support better maintenance of brain structure and function over time. Moreover, centenarians often display a favorable lipid composition in the brain, including higher levels of omega-3 fatty acids and lower levels of pro-oxidant lipids, which may help preserve membrane fluidity and reduce neuroinflammation. Diets rich in marine fats, vegetables, and polyphenol, common in many blue zone regions, are associated with enhanced cognitive performance and are likely contributors to long-term neural preservation. The gut-brain axis also warrants mention, as the microbiota may influence brain aging through immune modulation, production of neuroactive metabolites, and maintenance of the blood-brain barrier. In centenarians, gut microbiome profiles are often more diverse and enriched with beneficial taxa, such as Akkermansia mucinifila and short-chain fatty acid-producing bacteria. These microbes generate metabolites like butyrate, which have anti-inflammatory and neuroprotective effects, adding another layer of resilience. Importantly, psychosocial factors also contribute significantly to cognitive longevity. Centenarians frequently remain socially active, emotionally balanced, and mentally engaged. Lifelong learning, strong community bonds, and a sense of purpose correlate with both cognitive health and increased BDNF expression. Stress resilience, shaped by personality traits such as optimism, conscientiousness, and adaptability, may buffer the brain against the damaging effects of cortisol and sympathetic overactivation. Sleep quality, too, is often preserved in centenarians, and adequate sleep supports neurogenesis, glymphatic clearance, and synaptic remodeling. Genetics undoubtedly plays a foundational role. Variants in genes such as APO, FOXO3A, and SIRT1 are enriched in long-lived individuals and influence both systemic and neural aging. For instance, the APOE2 allele is associated with lower Alzheimer's risk and is found more frequently among centenarians than the E4 variant, which increases vulnerability to cognitive decline. However, the modest effect sizes of these genes suggest that lifestyle and environmental interactions are essential for realizing their protective potential. While not all aspects of brain aging are avoidable, the lives of centenarians reveal that cognitive decline is not an inevitable consequence of growing older. By maintaining favorable metabolic, immune, and vascular conditions, engaging in enriching social and intellectual activities, and possibly benefiting from protective gene variants, these individuals exemplify the potential for sustained cognitive function into the second century of life. As the global population ages, translating these insights into public health strategies and clinical interventions becomes increasingly important. Whether through enhancing BDNF signaling, targeting neuroinflammation, preserving vascular health, or supporting lifestyle choices that reinforce brain resilience, there is growing hope that the lessons from centenarians can be applied more broadly to promote neuroprotection and cognitive vitality throughout the lifespan.